Today's notes are about the scientific method, or what we sometimes call scientific investigation. So what I want you to focus on today are what are some common steps that scientists use to solve a problem. When scientists are trying to solve a problem, we typically undergo a process that we call the scientific method. In the scientific method, there are a bunch of steps that scientists need to follow in order to answer this question or solve the problem that they have. The first step is identify the problem or question. And often this can be found by observing situations around you. Okay, so if you see something out in the open and you wonder how does that happen or why does that work, that's how you're going to be able to form your question. Or if you stumble upon something that's not clear, that's going to be your problem. So to start the scientific method, you need to identify the problem or question that you plan to investigate. Once you've done that, you can move on to step 2, where you're going to collect background research. When you collect background research, you're going to gather information to help you understand that problem or question. This might be information to help you understand how to set up an experiment or to help you understand some of the variables that might be involved. The research you find can be from a book, encyclopedia, or the internet. But one big key that you need to think about when you are researching things on the internet is whether or not these websites are reliable, whether or not the book you're reading is reliable. Some things that might help you is any website you find that has .edu, .gov, or .org is going to be a little bit more reliable than, say, a Wikipedia site. Once you have all of the background collected, you can start to form a hypothesis. A hypothesis is often referred to as an educated guess or prediction, and it's often written as an if something, then something statement. So if this happens, then that will happen. These are called educated guess because educated is coming from you getting your background research, and guess because you are predicting what is going to happen. Once you have your hypothesis created, you can actually start to design your experiment. And the big thing here is that you're going to create a very detailed procedure about how to test the hypothesis you just came up with, how to test that question. So when I say detailed, I mean very detailed. Let me give you an example of a not so detailed experiment or procedure. If I am making a peanut butter and jelly sandwich and I ask for you to give me a procedures list, if you say, grab a slice of bread, put jelly on that piece of bread, grab another slice of bread, put peanut butter on that bread, put the two slices together and enjoy, you gave me a procedure set. However, it's not the most detailed. We can make that more detailed. Why would we have to make it more detailed? That's in order for someone to be able to copy what you did and repeat your experiment. So for example, you might say, grab a slice of bread and put 2 tablespoons of jelly on the left side. Put 2 tablespoons of peanut butter on the other slice of bread and put them together. So the more detailed you are, the better your experiment will be. Now, when you're designing experiments, you're also going to have to identify some variables. The first variable you'll need to identify is called the independent variable, or the IV. And this is what is changed by the scientists. The other variable is the DV, or the dependent variable. And this is the variable that is affected by the independent variable, about what you changed. For example, if I wanted to see how much water or different amounts of water affected plants' growth, my IV would be the amount of water. That is what I am changing as the experimenter. I changed the amount of water, that's the IV. The DV would be how tall that plant grew. That's what I'm measuring. The height of that plant is going to be affected by the amount of water I give that plant. 2 more variables we're going to have to cover are the control, which is the group that remained unchanged, or the comparison group. So maybe the plant that got 0 water would be our control. And the second is constants. these are all the other factors that are kept the same. So for that example about the water and the amount of water and the height of the plant, the constants would be the amount of sunlight would be the same. We don't want to change the amount of sunlight because we're only changing the amount of water. We're going to make sure it gets the same fertilizer, that it's the same type of plant, that it's all the similarities outside of the IV need to be the same, or what we call constants. Once you've designed your experiment, you can move on to the fifth step, where we're going to collect and analyze our data. So once we've set up our experiment, we're going to measure and record these results. And we're going to record these results in a data table. So you'll want to make sure the data table is set up before you begin your experiment. Then you are going to usually take that information that is in your data table and put it into a graph that can visually represent what you found. It's easier for scientists to look at the graphs versus data tables to be able to notice any trends right away. Once you've collected and analyzed your data, you're going to draw conclusions, and there's going to be one specific question you're going to ask yourself. Does the data in my data graph, in my data chart and graph, does that data support the hypothesis? If it supports the hypothesis, you might say why it does, or are there any further questions that you have? What else could you change? If it does not support your hypothesis, you might answer why it didn't support your hypothesis. Are there any errors that might have happened in the experiment? Once you've drawn your conclusions, you get to move on to number 7, which is a big one. Report your results. Scientists need to be able to report their results so that other scientists can take that information and verify it. They're going to verify that information by repeating your experiment to see if they get the same results that you did. That is why the detailed procedure list is extremely important. Now, once you have completed your results, if you notice, it says that now you can reevaluate. So if your data supported your hypothesis, you can move on to a different experiment that might test a different IV related to the same thing. So instead of amount of water, does the amount of sunlight affect height of plants? If it does not support your experiment, or your hypothesis, does not answer your question, okay, you might want to think about why, or could you change something to help make that experiment worthwhile? And you'll start all over. Now I'm going to give you an example of the scientific method done with plants and fertilizers. So our experiment question is how to determine the effect of fertilizer on plant growth. So does different types of fertilizer help plants grow taller? So our independent variable, or what I change, the IV, would be the type of fertilizers. Say I have 3 different types of fertilizers, Scott's, Miracle, and organic fertilizer. I want to see which one of those will help the plant grow the tallest. So my dependent variable, or the DV, will be the growth of the plant. How tall does that plant grow? The control, which I said was the comparison, would be the plant that does not receive fertilizers. So as you can see at the bottom of the screen, there are 4 different groups. There's the control group, which will be the comparison group. It gets no fertilizer. And then the 3 types of fertilizer that I have. The constants in this experiment would be types of plant, because all the plants have to be the same. The amount of water, all 4 of the plants will get the same amount of water. They will get the same amount of sunlight and the same amount of fertilizer, but not type of fertilizer. The type of fertilizer is our independent variable. Then we will do our experiment, and we will be able to compare, let's say that between the Scott's, the Miracle, and the organic fertilizer, the Miracle fertilizer grew the tallest. Then we will compare all 3 of those with the control to see if fertilizer does in fact help grow plants. Now that we have finished our scientific method question, I'd like you to think and summarize what the purpose of the scientific method lecture was. Why do we need to learn about this? And I'd like you to answer the questions. Are there any topics you would like additional review of, or are you confused about a certain spot? Make sure you complete the notes on this and come to class ready to practice.